Good morning, everyone. So it might be the last online class uh, from Sunday onwards. We'll meet on the lecture hall. So on previous classes on the endocrinology, we have studied this one hormones. So mechanism actions of hormones, and is a hormone secreted from this pituitary gland. This hormone secreted from this thyroid gland, parathyroid gland. So from today onwards, we'll study. We will discuss on the hormone which is secreted from a pancreas. So from pancreas, basically it is three major hormones are secreted. So one is that is the insulin. So which is around this pancreas has a two types of it have two types of cells. One is that two types of functions. So endocrine functions and the exocrine function. So both functions are equal important for the metabolisms of a different these macromolecules. So exocrine functions, that means it secrete different this one enzymes that is necessary for the digestion of this carbohydrate, uh, in this protein and lipids. Similarly, that it also have what is the endocrine functions, so which regulate the metabolism of this carbohydrate, protein, and lipid. So on that, these endocrine functions, that 60 to 80% of these endocrine cells, that is, it have a different these cells, endocrine cells, that is alpha, beta, gamma. So alpha, beta, or delta. So this beta cell is responsible for a synthesis of insulin. So this insulin is synthesized from this Langerhans of the islets. So that these are the cells that is present on the pancreas, and this around 60 to 80% of these entire these endocrine cells, they are responsible for the synthesis of insulin, and 15 to 20% of the pancreatic endocrine cells, they are responsible for the synthesis of the another hormone called as a glucagon, and around 5 to 10%, so they are responsible for the synthesis of the another hormone called as a somatostatin. So this the actions of this insulin and the glucagon, so on the different metabolism is just opposite to each other. So which we'll discuss on the later class. So today we'll focus on the this insulin. So like these other hormones, so which we studied during this hormone synthesized from this pituitary organ. So these hormones, the hormone which is synthesized from pancreas, so they are also polypeptide in nature. So polypeptide in nature means that they are protein in nature and synthesized from a number of amino acids. So this insulin, so it is derived from a Latin word called as insula, and this insula literally mean it as a island. So generally these hormones, these it's synthesized from a the, so isolates of Langerhans, and this hormone, that is insulin, can be obtained highly purified from pancreatic extract, and this insulin is the first protein whose amino acid sequence was determined. So for this work, this Sanger, he got a Nobel Prize. During a synthesis of this insulin, it is synthesized as a linear single polypeptide chain. However, for the biochemical functions, it forms a two subunits, which is interlinked by a disulfide bond. So the origin of this both subunit is from a single polypeptide chain. So it consists of these two polypeptide chains. So chain A, that composed of 21 amino acids, and chain B, it composed of 30 amino acids. And these two chains, two polypeptides, they are linked by a two disulfide bond, which is made by a cysteine residues of each polypeptide chain, and this disulfide bond is found at the positions 7 and 20 of a chain, to the positions of 7 and 19 of the beta chain. So the molecular weight of this insulin is 600 Dalton. And apart from that, the A chain of the polypeptide, it has a intra-disulfide bond in between the cysteine molecules at position 6 and the 11. So this structure, so it is it's the structure of this one pro-insulin. So during a pro-insulin, what happens? It is a linear structures. So on that, this amino terminal, it forms a B chain, and the last carboxy end, it forms a A chain, and this the middle sequence, it is called as a connecting peptide, and during post translation modifications, this connecting peptide, so it is hydrolyzed, and so the fully active, biologically active this insulin is formed. And for the biologically activity of this insulin, both subunits are equal important. So on the hydrolysis of this sulfur bond by the alkali, it becomes a denature. That means it loses its biological activity. So both for the biological activity, it is both A chain and B chain is necessary. And this during a proteolysis, partial hydrolysis, this in a Golgi apparatus, what happens is both this insulin as well as the disconnecting peptide chains, they are present, and the functions of disconnecting peptide is till now it is unknown. So this, generally these cysteine molecules, so they are responsible for formations of disulfide bond. So they are responsible for to form a inter-disulfide bond. So that is the inter-disulfide bond, and this is the intra-disulfide bond, which is present in the polypeptide A. So in between 6 and 11. So in the pancreas, this insulin is often found as polymers. So that means they are present in a as a dimer or hexamer. So which contain a zinc. So if dimer hexamers means that is a 3 dimers. They are bind with 2 molecules of zinc and the 1 molecule of calcium. So during a storage, so storage found in the pancreas. So there are differences in the primary structures of insulin from a different species. So according to the species to species, the presence of the sequences of amino acids on the insulin is different. So that is a varied. However, also the same activity when administered to the individuals of a different species. So because of that, what happened? Before the preparations of the insulin, nowadays what happened due to the recombinant DNA, so we can get this one human insulin directly for the treatment purpose. Before that, this porcelain, this insulin, they are generally used in the human beings to reduce the blood glucose. So they have a different varied in the structure sequence of amino acid, but they are equally active in the different individuals. So individuals of a different species. So by synthesis, this human insulin resides on the gene which is responsible for the synthesis of this insulin, that is resides on the short arm of chromosome 11. So unique set of this one transcriptions factor found in the beta cell nucleus. This activates the transcriptions of the pre-pro insulin mRNA from the insulin gene. So during a transcriptions, so there is the pre-pro insulin mRNA is formed, and from that, after the translations, that is the pre-pro insulin is formed, and from that, what happened, like in the other proteins, what happened, the signal peptide from the N terminal, so that is released, so which composed of 23 amino acids. So during a when it is a free proinsulin, so generally it contains 109 amino acids, and from that this sequence signal peptide that is removed as soon as this insulin it enters to the endoplasmic reticulum. So which is of a 23 amino acid residues, and in endoplasmic reticulum, so it is converted into the proinsulin. So after the removal of these 23 amino acids, so which is called as a signal peptide, then this pre-proinsulin is converted to proinsulin, and this proinsulin, so it is transported to the Golgi apparatus. So where it is cleaved by a protease called as a prohormone convertase 1 and 2 in the two sides, so that what happened? This insulin and connecting peptide, so they are separated. So this connecting peptide, 33 amino acids is removed, and extra 2 amino acids are removed by the carboxypeptidase H. So now the biologically active insulin has a 51 amino acid sequences. So A has a 21 and B has a 30, so total 51. So this is a structure. So this structure for the synthesis of the order proteins also the mechanism is same. So that is this mRNA is synthesized in a nucleus. After that, what happened? By using that mRNA, these ribosomes are responsible for the synthesis of a protein, which is called as a translations. So as soon as this protein is synthesized, they enters to the endoplasmic reticulum, where this signal peptide is removed, and from endoplasmic reticulum, so they are transported to Golgi apparatus, where it is stored in the vesicles, and in the form of that vesicles, so they are transported to the targeted, so outside the this one cell membrane. So secretions. So the insulin is packed in granules in a Golgi apparatus. So during a packed, they both these vesicles, they contains both insulin as well as C peptides, and during a release also what happened, both this C peptide as well as the this insulin, it is released into the circulations. So generally, while they are stored in a this one pancreatic cells, that means in the vesicles, so they are associated as a hexamers. So with a 2 zinc ions and a 1 calcium ion, and these stored insulins, they are released by a exocytosis. For that, for the release of this insulin, which is stored in a pancreatic cell, so that is released in presence of a glucose. So that means a major stimulant is a glucose. So when we consume a food, so that food contains a carbohydrate. So after this carbohydrate, after the digestion, so what happened? This carbohydrate are converted to the monosaccharide, so majorly in the glucose. So that when this glucose is absorbed from a intestinal lumen, so this glucose, they are entered to the circulations, and from circulations what happened, this glucose, they enter to the pancreatic cell, and that glucose is responsible for the release of insulin, which is stored in a pancreatic cell. So I guess you all of you have discussed about this insulin released during your PBL session. So what happened? So on the when we in a well-fed state, that means after digestions and absorptions of a food, what happened is glucose is increased in the circulations, and this increased glucose, so they enter to the beta cells of pancreas, and here what happened in pancreatic cell, it contains a GLUT2 transporter. These GLUT2 transporters, they are only open when there is a high concentrations of glucose is present in the circulations. When there is a low concentrations of glucose presence in the circulation, so that time what happened, glucose, they do not enter to the pancreatic cell. So that's why what happened, these pancreatic cells are unable to release a insulin. So when there is a high concentrations of a glucose in the circulations, that time only this glucose, they can enter to the beta cells of pancreas via the GLUT2 transporter. And in beta cells, what happened, this glucose, so they oxidized to produce a ATP. So when there is a increased ATP, so that cause the closure of potassium channels in beta cells. So when there is the closing of these potassium channels, so that causes the depolarizations, and this depolarizations is responsible for the opening of this calcium channel, this voltage gated calcium channel, so that the calciums which is present in extracellular fluids, they enters to the beta cells, and these increased concentrations of, that is the increased concentrations of the intracellular calciums, so that is responsible for a release of insulin from a vesicles via the exocytosis. So these intracellular calciums that lyses the vesicles, which you have also studied during a release of acetylcholine during a neurotransmitter. So on neurotransmitter also what happened is calcium voltage gated calcium channels. So they are responsible for a releasing of acetylcholine, which is stored in the these vesicles. So similarly, here also what happened is insulins, which is stored in the vesicles. So that is released by the increased concentrations of the calcium. And for the opening of this voltage gated calcium channel, it require a increase in ATP concentrations, and this ATP concentration is increased by the oxidations of the glucose. For that, glucose should be entered to the beta cells, and which is usually done during a hyperglycemia, so that which takes place after a food consumption. So apart from this glucose, this cyclic AMP, it is also the one of the main modulator for insulin release. So as we discussed, what happened is intracellular concentration of calcium is responsible for the release of insulin from a beta cells of pancreas, and for that, the increase of this intracellular calcium, it is not only increased by the opening of this voltage gated calcium channel. You have studied this during this muscle contractions. What happens? Some of these calciums, they are also stored in the endoplasmic reticulum and the mitochondria. And these the calciums which is stored in the endoplasmic reticulums, so that is released by the another these activations of the another the second messenger, like this diacylglycerol and inositol triphosphates. So like that, this cyclic AMP, so that also they can increase the intracellular calcium concentrations, and this increased calcium concentrations in the cytosol, that may be the responsible for the release of insulin. However, when there is a low concentrations of the glucose, the increased calcium level by the cyclic AMP and the others, that is in the diacylglycerols and inositol triphosphate, they have a no role. So they do not affect on the release of a insulin. Though the cyclic AMP, diacylglycerols and others, they can also be the modulator of the insulin release, but in absence of glucose, they have no role. So they are not able to release the insulin. So sulfonylureas, so you have studied this drug, so which is generally used to treat this one diabetes mellitus. So this sulfonylurea, so it stimulates insulin secretions by closing the potassium channel. So when the potassium channel is closed, then what happens? There will be the depolarizations occurs, and so that this voltage gated calcium channel can be opened, and the increased calcium concentrations, now it can able to release the insulin. So sulfonylurea, so which is generally used to treat diabetes, so that act on the potassium channel, so which close the potassium channel. And simultaneously that the another drug, that diazoxide, so it inhibits the closing of potassium channel. Generally the diazoxide, it opens the potassium channels so that it reduces the these depolarizations and inhibits the insulin release. So apart from this glucose, this elevated level of amino acids, especially this arginine and lysine, and the free fatty acids, and also the GI hormones, they can promote the insulin secretion. And these GI hormones, so they are able to release the insulin. So that can be the best examples on that. When the person's, so there is the more insulin will be released after the food consumptions, then this intravenous supplementations of the glucose even in the same concentrations. So if we give a suppose if we just provide this 100 milligram of this glucose by the intravenous, then that increase this glucose is able to release the insulin. On the same time, the same if we provide 100 milligram of glucose via the uh, oral routes. So that time what happened? It estimates the more amounts of the insulin secretions than the these 100 same amounts of glucose which is given by the intravenous. Because so when we provide a glucose from a oral, so that time what happened? The for the releasing of insulin from a pancreas, apart from the glucose, the GI hormones they also this. So when we take some foods, that what happens? The GI hormones they get stimulated, and these GI hormones they also aid on the releasing of insulin from a pancreas. So apart from this one, amino acids, arginine and lysine, free fatty acids, and GI hormones and the glucose, these they are they all stimulates the releasing of stimulates the secretions and the releasing of the insulin. And these alpha adrenergic receptors and the somatostatin, so it depress the release of insulin. And the somatostatin, so it is secreted by the delta cells of a pancreas itself. And these somatostatins have a inhibitory effects on the secretions and release of insulin as well as the glucagon. So under these basal conditions, the insulin concentrations in plasma is 0.4 to 0.8 nanogram per ml. So it is around 61 to 122 picomole per liter. And the concentrations of insulin in the circulations begins to rise after 10 to 15 minutes of carbohydrate rich food, and it reaches maximum at 30 to 45 minutes. So this insulin have a very short half-life, which is less than 10 minutes, and generally it is degraded by the enzyme called as a insulinase. This insulinase enzyme is present in liver, kidney, and the other organs and the tissues. So mechanism of actions. So it actions is depend on the tyrosinase kinase activity. So on the target tissues, there is a specific that is the insulin receptors are present. So these insulins, so they after the release in the circulations, so they are transported to the target tissues, and where on the surface of the target tissues where there is a presence of insulin receptor, on that specific receptor, this insulin they get bind. And generally these insulin receptors, so they are composed of the 4 subunits. So you can see here 4 subunits. So there is a 2 alpha and 2 beta units. So generally that these 2 alpha subunits, so they are present on the surface of plasma membrane, whereas these beta subunits, so they are embedded the plasma membrane, and then they are continued to the cytosols of a target tissue. So when these insulin they get bind on the alpha alpha subunits of the insulin receptors. So insulin receptors, so which is composed of 4 subunits, 2 alpha subunits and the 2 beta subunits. And these alpha subunits, they are present on the this one surface of the plasma membrane of the target tissue, whereas these beta subunits, so they are integral, this embedded on the plasma membranes, and then they are continued to the cytoplasm of a target cell. So when this insulin, it binds on the alpha subunits, so it changes the conformations over the beta subunits. And generally what happens is beta subunits, it have a tyrosinase kinase activities. So that's why what happened when the insulin, it binds on the alpha subunits, there is the conformation change occurs on the beta subunits also. And during the conformation change to beta subunits, what happened, the tyrosines which is present on the beta subunits, so they get a phosphorylated. And this, when this tyrosine get phosphorylated, now this beta subunit, it's get activated, and this active beta subunits, it phosphorylates the another the proteins which is present to the which is present near to that beta subunit. So generally these proteins which is present on near to this beta subunits, so they are called as a this IRS, insulin responsive substrate. So which gets phosphorylated. So when this insulin receptor substrate, so they get phosphorylated, now it activates the another this one another enzymes like this one, this uh, GRB2, PI3K. So from which the sequence of the reactions takes place, and then it activates the targets enzymes which is present on the in the cell. And this insulin, it shows as the physiological functions to that target cells. So which you can see on this language also, that on binding of insulin on insulin receptor, so the conformational change takes place on the beta subunit, so which cause autophosphorylations and acquire the capacity to phosphorylate tyrosine residues of a protein that contains SS2 domain, so which the bind to receptor tyrosine residue. So these proteins, so which contains a SS2 domain, so the best example is that is the insulin receptor substrate. So that you can see on this figure also. So what happened, the binding of insulin on the insulin receptor which is present on the targeted tissues, so it get conformational change and which get also autophosphorylations over the tyrosine residues of these beta subunits, and which activates the another proteins which contains a SS2 domain. So best example is that is the insulin receptor substrate. So there are different insulin receptor substrates presence over the cells. So there are 4 different. So that is the IRS 1 to 4. Among that, there is IRS 1 and IRS 2. So they are majorly distributed on the most of the tissues, and it is found more in a liver and muscle. Like that, this IRS3, so it is predominant in adipocytes, fibroblasts, and hepatocytes. And this IRS4, so it is generally present on the embryonic kidney. So apart from that IRS, that's the insulin responsive substrate, uh, at least order 5 proteins have been identified to bind to insulin activated receptors. So once phosphorylated, the tyrosine residues in IRS to other protein containing SS2 domains, including the phosphoinositol 3 kinase and GRB2, so which continue the cascade of signaling events in the cells. So other pathway downstreaming of IRS include the phospholipase C gamma and the PKC protein kinase C. And I hope that most of you are familiar with these terms. So that is a phospholipase C protein, this one phosphoinositol 3 kinase. So phospholipase C that acts on the phospholipids and which produces a 2-second messenger, that is the diacylglycerol and inositol triphosphate. And generally these second messengers, so they activates the protein kinases. So for this one, cyclic AMP, for remembering the cyclic AMP, it activates the protein kinase A. And this phosphoinositol 3 kinase, it activates the protein kinase B. And this diacylglycerol and inositol triphosphate, so they generally activates the protein kinase C. And these unturns, these protein kinase enzymes, so they phosphorylates the proteins. So on phosphorylations of that proteins, they get activated and they shows the functions. So this is the picture from which you can see the series of the reactions. So after the binding of insulin to the insulin receptors, it gets activated the related proteins. So that IRS, this one insulin related substrate. And on activations of this insulin, this relative substrate, it activates the another proteins called as this PI3, K, and this GRB. So that is the growth related bound growth factor bind protein. So on activations of this growth factor binding proteins, so it releases it activates the cascades of the proteins, so which cause the activations of MAP kinase pathway. And this MAP kinase pathway is, it directly regulates the this one transcriptions of various proteins. And the activations of this PI3 kinase, it activates the protein kinase B, and this protein kinase B is responsible for activations of this GLUT4. And you may remember that this GLUT4 transporter, they are generally present on the muscle and the adipocytes. And this by which, due to the activations of this GLUT4, what happened, these muscles and the adipose cells, they are able to take the glucose from the circulation. So that's why when there is a release of the insulin, now it clear the glucose from the circulations, and hence what happened, so the glucose which is increased due to the consumptions of a food, now it is uptake by the cells, and then the concentrations of glucose over the on the circulations get maintained. So both are the same structures. So these are the different actions which is done by the insulin. So insulin can cause the protein translocations. So that means an enzyme activity, it can increases the activity of the enzymes, and it can increases the gene transcriptions, and it is also can involve in the cell growth. So major biochemical effects of the insulin is, so one is that is the activations of a factor, so that control initiation of protein synthesis. So it is the insulin is the anabolic hormone, so which is responsible for the synthesis of a protein. So for that, the amino acids should be transported to the targeted cell, so which is done by the insulin. So activations of transport of some amino acid into the muscle, and this amino acid is utilized for the synthesis of a protein. And apart from that, it also involved in the activations of a protein kinase, so which you have seen on the previous slide. So that means it is able to activations of protein kinase B, on which in turn that was what happens, the activations of protein kinase B cause the activations of GLUT4, that is the glucose transporter 4, and these translocations of glucose transporter 4 causes the transport of glucose from the circulations to the muscle and the adipocytes, where these adipocytes and muscles, they used glucose as the source of energy. So these muscles, they use glucose for the energy purpose, as well as it also used to synthesize the glycogen. And in adipocytes, this glucose is used for the energy purpose, as well as it is also used for the synthesis of a triacylglycerol. And it also causes the activations of cyclic AMP phosphodiesterase. And generally, these activations of cyclic AMP phosphodiesterase cause the inhibitions of the functions which is done by the glucagon. And apart from that, it also causes the activations of ribosomal protein kinase. You may be the familiar with this picture. You have studied this picture during these one regulations of glycogen metabolisms. So that is done by the glucagon. So here what you have seen, that is written when this glucagon and epinephrine, so they activates, then they are responsible for the synthesis of the cyclic AMP. So it is the second messenger. And on terms, so on activations of the cyclic AMP, that increasing of the cyclic AMP concentrations, what it do, it activates the protein kinase A, and this protein kinase A is responsible for the activations of this glycogen phosphor. So it causes the activations of glycogen phosphorylase. And the main function of insulin, so it activates the this phosphodiesterase enzyme, so which cause the degradations of cyclic AMP to the 5' AMP. So that what happened, there is the decreased concentrations of cyclic AMP on the cell. So which when there is a decreased concentration of cyclic AMP, then what happen? The actions of glucagon and epinephrine cannot be shown on the cell. So that's how this insulin, it inhibits the activity of glucagon and the epinephrine. So physiological effects of this insulin. So these main net effects of these actions on carbohydrate metabolism. So net effects of the insulin over the carbohydrate, it's lower the blood glucose level and increase the glycogen storage store. So for that, it perform a different work. So first one is that it increase the glucose uptake. So it induces the translocations of GLUT4. So this GLUT4 is responsible for the uptake of glucose by muscle cells and adipocyte cells from the circulations. When these cells uptake of glucose from a circulations, what happen? The concentrations of glucose on the circulations get decreased. So apart from that, it activates the glycolysis. So by means this glycolysis is, that is the oxidations of glucose. So from by which it increases the use of glucose as a source of energy. So for that, it stimulates the activity of glycolytic enzymes. So main key enzymes like this glucokinase, phosphofructokinase, and pyruvate kinase. Apart from that, this it also induce the synthesis of these enzymes, glycolytic enzymes like glucokinase, phosphofructokinase, and pyruvate kinase. And next thing is it increases the conversions of pyruvate to acetyl CoA. So by the which what happened, then this acetyl CoA, so that enters to the TCA cycles, and which is able to synthesis of number of ATP. For that, it activates the actions of this pyruvate dehydrogenase enzyme, so which converts pyruvate to the acetyl CoA. So it stimulates the glycogenesis. So this glycogenesis is, that is the synthesis of glycogen by using a glucose, and this glycogenesis is generally takes place in a liver and muscle. And the main key enzyme for a glycogenesis is, that is the glycogen synthase, and this insulin, it activates the actions of glycogen synthase. And apart from that, it also induces the synthesis of this enzyme glycogen synthase, and in the same time it inhibits, it inactivates the key enzymes of glycogenolysis called like this glycogen phosphorylase and phosphorylase kinase. And this insulin, it causes a decreased gluconeogenesis. So that is the gluconeogenesis, means that is the synthesis of glucose from a non-carbohydrate source. And for that, this insulin, it suppresses the activity of the enzymes like this pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase, and glucose 6 phosphatase enzymes. So it inhibits the synthesis of these key enzymes of the gluconeogenesis. So this pyruvate carboxylase enzyme. So which is the key enzymes that converts the pyruvate to oxaloacetate, and this phosphoenol pyruvate carboxykinase. So which converts the oxaloacetate to the this phosphoenol pyruvate, and this fructose 1,6 bisphosphatase enzymes, which converts the fructose 1,6 bisphosphate to the fructose 6 phosphate, and this glucose 6 phosphatase enzyme, so which converts the glucose 6 phosphate to the glucose. So decrease glycogenolysis. So that is the degradations of a glycogen. So it is done by the direct inactivations of the key enzymes of glycogenolysis, like this glycogen phosphorylase and the phosphorylase kinase. So it also increases the HMP shunt. This HMP shunt is responsible for the synthesis of NADPH, and this NADPH is necessary for the synthesis of fatty acids. So for that, it initiates the synthesis of this glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. And these both enzymes are the key enzymes for the oxidative pathway of this HMP shunt. So HMP shunt means that is a hexose monophosphate pathway, so which is responsible for the synthesis of NADPH, and this NADPH is utilized for the synthesis of fatty acids. So action on lipid metabolism. So net effects of this insulin over the lipid metabolism is it's lower the free fatty acids level on the circulations, and it increases the synthesis of triacylglycerol in the adipocytes, so which causes the store of this triacylglycerol. So for that, it causes the decrease lipolysis. So that means it decreases lipolysis, that is the it decreases the degradations of triacylglycerol on the adipocytes, and for that it activates the phosphoprotein phosphatase and phosphodiesterase enzymes, so which converts these TG lipase enzymes, that is the active TG lipase enzymes, into the inactive TG lipase. So triglyceride lipase enzyme, so that enzyme is the key enzyme, so which degrades triglyceride into the diacylglycerol, and then diacylglycerol to the monoacylglycerol and free fatty acids, and again this monoacylglycerol is the glycerol and the free fatty acids. So when these lipase enzymes hydrolyze the triglycerides, it is converted to the glycerol and free fatty acids, and this insulin, it inhibits the activity of triglyceride lipase, so that there is no degradations of triglycerides. For that, it activates the phosphoprotein phosphatase enzymes, so which converts the inactive this active form of TG lipase into the inactive form, and further it also inhibits the phosphodiesterase enzyme, so which causes the decreased concentrations of the cyclic AMP. So increased fatty acid synthesis. So it due to the increased availability of acetyl CoA and NADPH, what happened? This causes the increased synthesis of fatty acids. So for the this increased synthesis of fatty acids, so it activates the key enzymes of fatty acid synthesis, like this acetyl CoA carboxylase, so which is responsible for the conversions of acetyl CoA to the malonyl CoA, and it increase the activity of ATP citrate lyase enzymes, so which is able to the conversions of citrate to the acetyl CoA in the extra on the cytosols, and it also increases the fatty acid synthesis, fatty acid synthase enzyme. So increased fatty acid synthesis is done by this insulin, and for that it activates the key enzymes of fatty acid synthesis like this acetyl-CoA carboxylase, ATP citrate and fatty acid synthase complex. So which contains a number of enzymes. So it increases the synthesis of triacyl glycerol. For that, it increases the providing of this alpha glycerol phosphate. So when this glucose enters to the adipocytes, we have the glucose transporter for this glucose. So they enter to the glycolysis and they provides the synthesis of glycerol phosphate. And apart from that, it also induces the synthesis of lipoprotein lipase. And this lipoprotein lipase, it is generally present on the endothelium of the circulations. And this lipoprotein lipase enzyme is responsible for the degradations of a triacyl glycerol presence on chylomicron and VLDL. So the chylomicron, which contains the dietary fats, so it is degraded by the actions of lipoprotein lipase, and this after the degradations of these triglycerides presence on chylomicrons by the lipoprotein lipase, it releases the free fatty acids, and these fatty acids it enters to the adipocytes where these fatty acids are reutilized by the adipocytes for the synthesis of triglycerol. So triacyl glycerol. So due to the decreased level of free fatty acids, so there is a less beta oxidations and less acetyl-CoA is available for the synthesis of cholesterol and the ketogenesis. So there is the decreased ketogenesis. So actions on this on protein metabolisms. So net effects of this on the protein metabolisms, it promotes the protein synthesis. For that, it stimulates the uptake of amino acids by the different cells, and then it enhances the activity of these different enzymes, that is the RNA polymerase and these the enzymes which is responsible for the these translations. So that is the peptidyl transferase enzymes. So the actions of these enzymes is increased, and so that what happened, so it causes a protein synthesis. So that is the positive nitrogen balance. So that because of that what happens, so all the amino acids which is utilized by the cells for the synthesis of protein, so there is the less degradations of these amino acids, so which lead to the decreased productions of urea in the liver. So in presence of these insulins, so that's the increased insulin, so what happened, there is the decreased productions of the urea in the liver. So because the amino acids, they get less degraded, these amino acids are utilized for the synthesis of the protein. So since in presence of insulin, the glycolysis and the other carbohydrate metabolism is enhances. So because of that what happened, the maximum utilizations of these potassiums and the inorganic phosphate is takes place, so which causes the decreased concentrations of potassiums and inorganic phosphates on the circulations. So because these potassiums and phosphate, they are maximum utilized by the cells for the carbohydrate metabolisms and the protein metabolism. So clinical disorders of this insulin metabolism. So on that, these two disorders are there. So one is hypoinsulinism. So that is the low concentrations of insulin in the circulations. And next one is the hyperinsulinism. So that is the high concentrations of insulin in the circulations. So low concentrations of insulin in the circulation is generally related with a that is generally due to the absolute or relative insulin deficiency, and when there is the low concentrations of insulin on the circulation, so that lead to the diabetes mellitus, and this diabetes mellitus is the disorder where the hyperglycemia is seen. So due to the absence of insulin, what happened is glucose cannot be cleared from a circulation, so that lead to the diabetes mellitus. And this hyperinsulinism, so that is the increased concentrations of insulin in the circulation, and generally it is seen during a tumors of pancreatic beta cells, so which causes the uh, increased synthesis, uncontrolled synthesis of the insulin from the beta cells, and generally it is characterized by the hypoglycemic crisis. So when there is a maximum insulin in the circulation, so what happens? The more amounts of glucose is cleared from the circulation. So that lead to the hypoglycemia. So this tumor of pancreatic hyperinsulinism is characterized by the hypoglycemic crisis, weakness, intense sweating, and the dizziness. So this is all about the insulin, and this lecture is made on the references on this book. So that is a textbook of medical biochemistry.